Okay, so let's get started. Um, Hello, everyone. Welcome to our today's Immune Zoom seminar. So it's my pleasure to welcome our today's speaker, Dr. Leng Fangwei from Chinese Institute for Medical Research, Beijing, China. So Fangwei currently is assistant investigator in CIMR. So he completed his undergraduate study in biotechnology from Sichuan University, and then continued with PhD training in biochemistry and molecular biology in Institute of Biophysics, Chinese Academy of Science in Beijing. After Fangwei completed his PhD training, he moved to US, started his postdoc training in biological chemistry and molecular pharmacology at the Harvard Medical School, uh, Boston Children's Hospital, and then did his postdoc training with Dr. Sun He, who also gave us a really wonderful talk a few months ago from Harvard. So he was then promoted as an instructor from Boston Children's Hospital, and then from 2023, he decided to move back to China and start his independent research program as an assistant investigator at CIMR, Chinese Institute for Medical Research. Fangwei's lab actually systemically investigated the gene regulatory mechanism of Treg cells from the perspective of protein-protein interactions, three-dimensional chromatin architecture, and the epigenetic modifications. His lab also designed small molecules, peptides, and then nanobodies targeting key proteins such as FoxP3 to modulate Treg function, aiming to develop therapeutic strategies for autoimmune disease and cancers. The main research direction from Fangwei's lab, including mechanistic study of Treg protein interaction networks and the gene regulation circuit in immune homeostasis and disease. Second part is to develop of small molecules, peptides, and nanobodies that targets immune suppressive function of regulatory T cells. Last but not least, exploration of the biological functions of microsatellites DNA and its potential clinical relevance, which I think is going to be the main topic of Fangwei's today's talk. So I think without further ado, Fangwei, thank you for delivering lecture with us today, and then we are very much looking forward to your talk.
Okay, thanks, Dr. Wu, for the kind introduction. I have been following the Immune Zoom seminar since the pandemic and have learned a lot from here. And today I'm going to talk about our work on FoxP3, which is a key transcription factor in Treg cells. I will show how FoxP3 recognizes microsatellite to coordinate the 3D chromatin structure and how this in turn shapes the gene regulation network in Tregs. So since many of you are immunologists, so you might be familiar with Treg cells, especially given the recent announcement of the Nobel Prize awarded to Mary Brinko, Fred Ramsdale, and Sam Sakaguchi for their discoveries on the peripheral immune tolerance and how Treg cells prevent us from autoimmune disease. So just a quick refresher, let me briefly introduce the discovery of Treg cells. So all of this begins with the observation that removing the CIMRs from mice 3 days after birth will lead to autoimmune inflammation. As we know, the CIMRs is responsible for generating T cells. So at the beginning, people thought that if we get rid of the CIMRs, so the overall immune response should be reduced. However, the result was observed was the opposite. There is increased immune response. And Sam Sakaguchi and his team confirmed that transplanting the lymphocytes from normal mice to mice without CIMRs could reduce autoimmune reactions. So from then, he became convinced that there should be some suppressor T cells in the mice. And finally, in 1995, Sam Sakaguchi and his colleagues identified a subpopulation of CD4 T cells expressing CD25. so which is about 10% of the total CD4 T cells. So it shows remarkable ability to suppress the immune responses and was finally named as regulatory T cells in 2000 after confirmed by many other groups. So Brinkel and Ramsdale later discovered that the mutations on FoxP3 protein will lead to heavy autoimmune disease in scurvy mice and IPX syndrome in human. Another breakthrough which finally links FoxP3 and the Treg cells in 2003. And three different groups identified FoxP3 as the master transcription factor to drive the Treg development and function. So expression the FoxP3 protein can function or convert the normal T cells into Treg cells. So once it became clear that FoxP3 is essential for Treg development, research on this protein expanded very fast. So now we know that FoxP3 can function as both transcriptional activator and a repressor to regulate hundreds of genes. But the molecular mechanism remains unclear. And a key question that has puzzled the field for decades is that where does FoxP3 bind? And which genes are direct targets of FoxP3. So as a member of pocket transcription factors, FoxP3 was initially believed to bind the canonical pocket motif, so which is 7 nucleotide lengths. But it's very surprised that the enrichment of the canonical pocket motif in FoxP3 chipset peaks is only about 20%. And this is ranking even lower than many other transcription factor binding motifs. Uh, and most of the single focal motif sites are not occupied by FoxP3 in footprinting assay. So that's where I started my project, and we aimed to explore the direct targets of FoxP3 from a biochemical perspective. So here I will briefly introduce the domain architecture of FoxP3 protein. So the N-terminal domain is responsible for the cofactors recruitment. The coil-coil domain is for dimerization. And the RBR loop is a highly flexible loop with very limited information. So we named it as RBR because it can recruit the cofactor RUNX1. And I will show you more details about our findings on this loop region later. So the C-terminal domain, focal domain, is a highly conserved DNA binding domain, which is shared by around 50 human transcription factors. So it was previously reported that the truncated focal domain can recognize single focal motif as a domain-specific confirmation. However, as I mentioned earlier, so this model cannot explain the in vivo sequencing data. So we decided to investigate FoxP3 using our more native construct. So we successfully purified FoxP3 delta protein, which covering all domains except the intrinsic disordered N-terminal region. So to test the DNA binding, we did a literature shift assay. And when we incubated, increased the amount of FoxP3 protein with different DNA molecules, and we found that FoxP3 has very low binding affinity to the DNA containing single focal motif or tandem repeats of focal motifs. Instead of that, FoxP3 prefer to bind the inverted repeats of two focal motifs. And as you can see, this is the complex band. And consistently, full-length FoxP3 protein expressed in 293 T cell shows the same DNA preference in the DNA pull-down experiment. So the different DNA preference suggests that FoxP3 may function in a different way from the truncated focal domain. And this is further proved by our crystal structure of FoxP3 delta protein in complex with DNA, which turns out to be a head-to-head dimer. So this structure can explain perfectly why FoxP3 prefer to bind the inverted repeats of focal motifs. So when we compare one monomer from our structure to previous structure, we found them in completely different conformations. So in our structure, the RBR loop region can help to stabilize the long spike conformation. And when we get rid of the RBR loop, this hydrophobic patch will extend to form a long alpha helix, exposing the hydrophobic residues, which are labeled in yellow color here. And this residues can serve as an interface to form the domain spec dimer. So then you may wonder which of the two conformations is physiologically relevant. To address this question, we generated a mutation that can repress the domain spec conformation. And as a result, it has low impact on the DNA binding and the transcriptional activity of FoxP3. On the other hand, when we introduce the patient mutation, which is R337Q, it can induce the domain spec conformation and result in much lower DNA binding and the transcriptional activity of FoxP3. So interestingly, when we combine these two mutations, it can partially rescue the DNA binding and the transcriptional activity of FoxP3. So suggesting that the long spec conformation should be the functional form of FoxP3, while the domain spec conformation will result in loss of function. And this finding provided us new insights into the pathogenic mechanism of some IPX mutations, especially those mutations located in the DNA binding domain. And I think this will help us to develop some new therapy strategies. So it has shown, I have shown you that the RBL loop region can help to stabilize the long spec conformation. And the structure also shows that it has additional function to mediate the head-to-head dimer. And this feature is unique to FoxP3. So unlike the highly conserved the focal domain and the coil domain, the RBL loop region shows significant diversity among different FoxP proteins. As you can see in the literature shift assay, only FoxP3 can form dimer on the inverted repeats of two focal motifs. And as for the FoxP1, P2, and P4, they bind all DNA molecules as a monomer, no matter there is single focal motif or inverted repeats of focal motif. So this is very different from FoxP3 protein. So when we introduce the RBR mutation on the head-to-head dimer interface, as you can see, they have greatly impact on the FoxP3 transcription activity, DNA binding, as well as the cofactor recruitment when you compare the mutations to the wild type. So from the head-to-head dimer structure, we think the inverted repeats of the focal motifs should be very strong binding targets for FoxP3. So we wondered if this sequence is enriched in FoxP3 chipset data in T-Rex, but that's not the case. And we were kind of confused why the second FoxP3 binding set could not be detected from the global motif analysis. So one possibility is that FoxP3 may bind some other suboptimal sequences as the second binding site. So this is reasonable because there are several low canonical focal motif has been reported to be recognized by other focal proteins. For example, so FoxN3 has been reported to bind two completely different motifs. So indeed, when we combine this reported non-canonical focal motifs together with the canonical focal motif, and some of them shows very high binding affinity to FoxP3. To explore the FoxP3 preferred suboptimal sequences systematically, we perform the unbiased potency using degenerated DNA pools, which contain a random sequence on the second binding site or both sites. And when we use the pull down library for sequencing, and as a result, we found that besides the canonical focal motif, FoxP3 also has intrinsic high binding affinity to two major groups of sequence. So the first one share the TGTT core sequence, which is more similar to the canonical focal motif. And the second one share a GCAT core sequence, which is new identified sequences. So then we checked if FoxP3 can recognize genome locus with a combination of group 1 and group 2 sequences instead of perfect inverted focal motifs. And indeed, we found a lot of native FoxP3 binding sites harboring these two groups of sequences in head-to-head configuration. So here I just show some of the examples. Actually, when introduced a single nucleotide mutation on one of the FoxP3 binding sites, such as the C2A mutation and the A2T mutation here, there is significant reduction in FoxP3 binding in cut and run sequencing, as you can see here, and also in vitro pull down experiment. So this sequence is without mutation, and this is with mutation. So we proved that FoxP3 can recognize focal motif or other suboptimal sequences as hit-to-hit dimer. But there are still many FoxP3 binding sites cannot be explained by such sequences. And we are not satisfied with stopping here. So we used another unbiased strategy to pull down fragmented mouse genomic DNA. And as a result, the dominant enriched sequence is TNG repeats. Consistently, when we revisited the previous published in vivo chipseq data and the cut and run data from Densky lab, there is also enrichment of similar TNG repeats, so which has been ignored for some reason, probably because simple repeats are normally discarded as junk DNA during motif analysis. So when analyze the distribution of the TNG repeats, we found that nearly 38% of FoxP3 bound TNG repeats are located in the promoter region, and much higher than the ratio of genome-wide TNG repeats. So here is an example of long TNG repeats, which is located in the FoxP3 bound promoter region. So we also found a high level of activation and the promoter marker of FoxP3 bound TNG repeats, and also much higher chromatin accessibility, suggesting that the TNG repeats may play some important role in transcriptional regulation in Treg cells. So we moved on to ask how FoxP3 bind the TNG repeats using our chemistry experiment. So we focused on T3 repeats at first because it's the most enriched sequence in our pull down experiment. And the late feature shift assay suggests that FoxP3 can form a monomer on T3 repeats, but not on the inverted repeats of focal motif, which I have shown you in my previous slides. So lecture-style EM analysis of the monomer sample clearly show filament-like structure. As you can see, they're very clear filament-like particle. So we naturally thought that the FoxP3 hit-to-hit dimer may wrap around a single DNA molecule as hexamer to form the filament, as I shown here. But in part, this is in part because the her lab has many experience in working with RIG-I and MDF5 filament. But this model has misled us for a while until I finally saw the cranium structure. And here is the more clear model of the apple structure. So in this structure, it's very surprised that FoxP3 can form a laser-like monomer to bridge two DNA molecules. As you can see in this model, each FoxP3 monomer will recognize 7 nucleotide motif, which is TG, TTT, GT. It's very similar to the canonical focal motif. There are only 2 nucleotide difference between them. So the structure strongly suggests that FoxP3 may play some important role in forming DNA looping, as the model shown here. To look at this, we analyze the Plexic data, which can provide us information about chromatin interaction. As a result, we can find a high ratio of chromatin interaction between two TNG sets. And this is the ratio colored in orange color. So and the ratio is even higher than 50% when we look at the strong chromatin interaction. Considering the genome-wide frequency of the change repeats is only 0.06%, so this is a very high enrichment. The importance of the laser-like monomer was further confirmed by generating mutations on the monomer interface. So when compared to the wild type, those mutations has a great impact on the cellular function of FoxP3 as measured by the transcriptional activity suppression activity and the RNA-seq, and also TNG repeats recognition in the chipset data. So to check the directed contribution of the TNG repeats in FoxP3 binding, we also analyzed the FoxP3 cut and run seq data using Treg cells from B6 and the cast hybrid mouse. So since there are enough difference between the B6 and the cast genome sequence, so the cut and run data can tell us which sequence is preferred by FoxP3. So for example, there are more reads mapped to the B6 genome at this locus, suggesting that FoxP3 has higher binding to the upper sequence. On the opposite, FoxP3 has lower binding affinity to the upper sequence because there is less reads mapped to the B6 genome. So based on this concept, we went through top 100 B6 or cast BIOS peaks and found that there are 47 of them show significant TNG deletions, suggesting that the FOXP3 binding affinity was determined by the length of the TNG repeats. And over half of them are T2G deletions. And here I just show one example of the T2G deletions. So in this locus, the cast genome sequence has much longer TTG repeats and shows much more FOXP3 binding in both the in vivo cut and run seq data and the in vitro pull down experiment. So actually, there are only 21% of FOXP3 or of T3 repeats are located in the FOXP3 cut and run peaks. And there are also high percentage of T2G and T4G repeats. So we ask another important question: how does FOXP3 bind other types of TNG repeats? To address this question, we did a lateral gel shift assay for 3 different TNG repeats on the same gel. As you can see, FoxP3 can also multimerize on T2G and the T4G repeats. And it seems that FoxP3 can form much larger complex on T2G complex repeats. So indeed, when we solved the fellow cryo structure, the T2G complex contains 28 FoxP3 monomers and 4 DNA molecules in a cylinder-like architecture, which is much larger than the T3G complex, which contains only 10 FoxP3 monomers and 2 DNA molecules. As for the T4G complex, it's even more complex because we can identify the 11 FoxP3 monomers to bridge 3 DNA molecules. So let's look at the detail of the T2G complex structure. So we can find the 2 different DNA bridging modes, the mode 1 between DNA A and the DNA B, and the mode 2 between DNA B and the DNA C. And here I show the density, cryo density of the mode 1 and mode 2. So as you can see, we can find a much stronger interaction interface in the mode 1, suggesting a more stable DNA bridging, which might happen first and serve as a scaffold to recruit additional DNA molecules. So there are also 2 DNA bridge modes in T4G complex. So the left one in anti-parallel mode and the right one in parallel mode. So it's interesting that FoxP3 can bridge DNA molecules in so many different patterns. So we asked why there is such a heterogeneity. And we found that the heterogeneity is mainly driven by the unique RBR loop. So now we come back to the RBR loop again. I have shown you that the unique function of the RBR loop in mediating the head-to-head dimer and the subsequent recruitment of the cofactor RUNX1. And here we found that the highly flexible RBR loop can also drive the adaptive multimerization of FoxP3 on different TNG repeats. And the RBR loop has been highlighted in different color here because they are from different DNA bridging complex. So when we overlap FoxP3 monomers from different complexes, the orientation of the RBR loops are completely different. My understanding is that the intrinsic flexibility of the RBR loop will enable FoxP3 to bridge DNA molecules with different distance and orientations. So the high heterogeneity of FoxP3-induced DNA bridging may make you wonder whether this complex is dynamic and not stable. By contrast, we found that this is ultra stable. To examine the stability, we performed a single molecule Ferretti experiment. We firstly tested the 2 DNA bridging by using a long chuck DNA construct, so which contains a Psi3 labeled TNG repeats and the Psi5 labeled TNG repeats fused by a linker. So before we add the FoxP3 protein, the Ferretti efficiency is very low, as you can see on the upper panel. But upon adding FoxP3 protein, so there is a significant increase of the Ferretti efficiency, as you can see. When we add 200 nanomolar FoxP3 protein, so there is a great shift of the Ferretti signal. And this is so this suggests extended confirmation of the long-chuck DNA. And also this FoxP3 induced DNA bridging is rock solid because when we watch the DNA bridging complex with buffer for 45 minutes, there's very limited change in the first signal. So based on the 2 DNA bridging experiment, we added a third DNA, which is labeled with Cy7 fluorescent, to see if 2 DNA bridging complex can serve as a scaffold to recruit additional DNA molecules. So here is the final result. So the x-axis shows the time of the wash step, and the y-axis shows the fraction of Cy7 fluorescent labeled DNA bound to the DNA bridging complex. And it shows that the pairwise DNA bridging complex can recruit Cy7 labeled TNG repeats DNA, but not the DNA with a random sequence. You can see the difference between the upper group and the lower group. Again, this complex is ultra stable even after 45 minutes wash. So to further quantify the number of DNA molecules in each complex, we performed a single molecule pull-down assay. We incubated the fluorescent and biotin-labeled DNA with FoxP3 protein to form the DNA bridging complex and applied to biotin pull-down. And then we can count the number of DNA molecules by counting the photobleaching steps. Here I just show some example of the 3 steps and the 4 steps, which indicates the 3 and 4D bridging complex. So here is the final result. So we can see FoxP3 can bridge 1 to 5 DNA molecules across all 3 types of TNG repeats. So it's worth to note that for T2G repeats, which is labeled in blue color here, FoxP3 prefer to form higher order monomers compared to T3G and T4G repeats. So this finding is consistent with our previous observation from the gel shift assay and the triome structures. So we also designed the luciferase assay to confirm if FoxP3 induced DNA bridging can regulate transcription. So normally the enhancer element and the promoter element are close to each other on the same plasmid to promote the luciferase expression. But in our experiment, so they are designed to be separated into 2 plasmids. We name that as enhancer plasmid and the reporter plasmid. And both elements are fused by TNG repeats. And it can, in our original design, if FoxP3 can bridge the TNG repeats, they can also bring together the enhancer and the promoter elements. So this can subsequently promote the expression of the luciferase. So as a result, we can see a significant increase of the luciferase expression in the presence of wild-type FoxP3 and only when both plasmids contain TNG repeats. So, this is the empty vector and the FoxP3 mutation control. So when we check the location of FoxP3 Cartier run peaks and the ATAC peaks, it's surprised that FoxP3 occupied TNG repeats as shown here. I'm not sure if you can see it clear. Um, so as shown here, they are often clustered and located near the edge of the open prompt region, which is the red color labeled peak. Uh, as shown in this locus, the upper is the cut and run peak, and the lower is the attack peak, and there are four cluster tinge repeats. So since leukotoxin positioning sometimes lead to lower prompting accessibility, so we are wondering if the leukotoxin plays some role in FoxP3 binding and the DNA bridging. So we tested the TNG repeat sequence located in both leukotoxin DNA and the flanking sequence. So in the first case, we reconstitute the leukotoxin with DNA containing different sequences. This is the leukotoxin band. And so when we add the increased amount of the FOXP3 protein and the depletion of the leukosomal band indicates FOXP3 binding, and this only happens when the leukosomal DNA contains the TNG repeats, but not any other DNA. And for the second case, when testing the TNG repeats located in the flanking sequence, we also observed the depletion of the leukosomal band, and it surprised that FOXP3 preferred the leukosomal DNA over free DNA. As you can see, the leukosomal band disappear at much lower FOXP3 concentration, suggesting that the leukosomal probably can facilitate FOXP3 binding and DNA bridging. So we further did a lative gel or electric scanning EM to check the FOXP3 induced DNA bridging in the presence of leukosomes. So the left is the free leucosome. So you can see the classic round shape of the leucosome. And the middle is the ladder-like DNA bridging complex without leucosome. And the right is the DNA bridging complex in the presence of leucosome. You can clearly see the leucosome part, which is in round shape, and the DNA bridging complex part. So our findings suggest that FOXP3 can bridge multiple genome sites to coordinate the 3D chromatin structure, and the leucosome can promote the FOXP3 induced DNA bridging by bending the DNA. So, consistently in recent years, more and more studies prove that FOXP3-associated chromatin interactions are essential for Treg function. So I have shown you that FoxP3 can bind to two types of motifs, the head-to-head motifs and the TNG repeats. So you may wonder if there's some correlation between them. So we also check this. Interestingly, when we checked the distribution of these two types of motifs, most of the head-to-head motifs are located nearby our TNG repeats. So here I was just to show two examples of them. So this happens more when the TNG repeats is shorter than 40 base pair. Probably because when the TNG repeats is long enough, so the FOXP3 binding our abilities are already high enough. But if the TNG repeats is too short, FOXP3 may lead the head-to-head motifs to help the binding of FOXP3 and DNA bridging. So we also check the ratio of head-to-head motifs and TNG repeats overlap. So in FOXP3 chipset peaks, over 80% of the head-to-head motifs are overlapped with the TNG repeats. But the ratio in the open chromatin region is only 25%. So suggests the head-to-head motif may promote the FOXP3 binding to short TNG repeats. So this is further confirmed by our in vitro native gel shift assay. So we tried a different combination of the head-to-head motifs and the TNG repeats. So only when placing the head-to-head motifs and the short TNG repeats in the same direction with very small gaps, smaller than 10 base pair, we can observe the clear multimer assembly of FOXP3 protein. So altogether, we found that FOXP3 can form monomers to bridge DNA and non-TNG repeats directly And at those loci with shorter change repeats, FoxP3 lead to bind the head-to-head motifs first, and which can serve as a seed to promote the forming of the DNA bridging complex. So upon forming different functional assemblies, FoxP3 can recruit different cofactors to control the development and function of Tregs. So MindLab in the CIMR will explore the interactome of FoxP3 and develop drugs targeting the functional FoxP3 assemblies and also its cofactors to modulate Treg activity. At the meantime, we are also interested to explore other master regulators in CD4 T cell development. So we're still recruiting members. So if you are interested in our directions, please feel free to contact us. And at last, I want to thank Professor Sangher and TJ Har and also all collaborators, especially Wenxiang and Dylan, and also all members from the Her lab. And thanks for your attention. I'd like to take any questions.